Welcome back to part 2 of immune responses and COVID-19. When we put out the first part of this video, there were an awful lot of folks who responded and who enjoyed the video because of the content that was involved. And so I thought I'd do a part 2 that addresses why it's necessary to get vaccinated even if you've had COVID, or what happens if you've been vaccinated and you get COVID later. So if you'll bear with me for just a couple of minutes, I want to do a quick review of the first video, and then we'll move on and talk about the topics for the day. So let's start with a little review. You will remember that all of humans interact with a whole lot of different things in our environments in something that looks like the bell curve. And you'll remember that most of us are somewhere here in this middle part of the bell curve in terms of our response. So you could look at this in a variety of different ways. You know, the height of people, if 7 foot is way over here and 2 foot is way over here, most of us are in this range of normal. And so going up on this axis, in this case, is the number of people, and down on the bottom is the height, ranging from about 3 foot to about 7 foot over here. And so most of us fall somewhere in here. This is the bell curve. This is what's normal. So when we talk about vaccines, we're talking about trying to stimulate an immune response that will be here inside this bell curve. Most people will respond in this way. Some people will respond extraordinarily well. Some people will respond less well to a vaccine. So that's what we're talking about in terms of what that looks like. And you will also remember that when we look at the immune response, we talked about a primary response and that we get a little bit of antibody, and in this case it's mostly IgM that's produced. This is that time zero. Time zero is here, and then this is 5 to 7 days after you're first infected or vaccinated with something. You get a little IgM and it starts to fall back off. You get that secondary response here where you see the vaccine for the second time, or you get infected for the second time, and then you see this huge immune response like this that's dominated by IgG. IgG is the antibody that you want here, which also begins to fall over time. And this is time down here on this axis. So it begins to fall, and gradually it falls even more. So this is the generalized immune response. What we're finding for COVID, for many people, is that when you get that second dose of vaccine, you get this nice peak, but then a lot of people seem to be dropping off a little more rapidly in their IgG response, and it's coming down to this resting level that's pretty low. So if you're a person who's been to the doctor and asked them to check your antibodies or check your titer for the vaccine, we're seeing a lot of folks down here in a titer range of 8 to about 64, which is pretty low. This number can go up into the thousands, so that's pretty low, which is why right now the CDC is recommending that after the second vaccine, if you wait 6 months, for many of us, it's good to get that booster out here. All right, and the booster is a third shot that would then stimulate that immune response. So here we're looking at a new peak. It would stimulate it here. It would actually go higher than this one and then track out a lot longer. So in time, it goes longer and it comes down. And here in the third response, again, it's mostly IgG, but we actually start to see a shift to some IgA. And this is the one that we really want in the third one, because IgA is the antibody that protects the mucosal surfaces like the nasal mucosa and the ocular mucosa. This particular antibody is the one that's protective against the infection itself. IgG and IgM, while they can protect sometimes against the infection, more often you might get a breakthrough infection and have a really mild case of disease, but then not get the severe symptoms because you have that immune response. All right, so that's it by way of background. Now I'm going to pause for just a second, erase the board, and I'll come back and talk a little bit more about why you need to get a vaccine even if you've already had COVID. So what we're going to talk about next is the difference between the vaccine and having a natural immune response. So coronavirus is a spherical shaped virus that has a couple of pieces of RNA in it. And this is RNA, both of these. You probably remember from your biology class that most organisms have DNA, and then there's this whole DNA to RNA to protein thing. Well, coronavirus is different. So it has this RNA inside, and it codes for a variety of different proteins that are then expressed on the surface. And I'm going to try to draw these with different shapes, so you'll get the idea and kind of hold on to it, that these are different things. And then one more. So I'm not going to label all of these, but I am going to draw your attention to one of them. And the one I'm going to draw your attention to is the one that is shaped like a box right here. And we're going to call this the S protein. And this is the one that's really interesting in terms of the vaccine, because both the Moderna vaccine and the Pfizer vaccine are based on the S protein structure. So if you look, I've drawn these in different shapes for a very important reason, because when your immune system looks at something, it looks at it solely in terms of shape. So the goal of the immune response is to be able to recognize the shape of this S protein. So I'm going to come over here and show you what I mean in a slightly larger diagram. So if this is the shape of the S protein right here, and I'm just going to squiggle in it just so you get the sense that it's filled in. All right, what the immune response does is it generates an antibody, and antibodies are shaped roughly like this. They're shaped like a Y. And then out here on the end, they have a binding region. And if you'll notice, the part of the antibody that will attach, so this is binding up here, this part is shaped in a complementary way to this. So if we were to flip this around and draw it, it would look something like this. And then there would be another one over here that would bind to that, and then the antibody goes up here like this. So this is the tail of the antibody, and you can think of this binding region as arms. And if you want to, those are hands. All right, now notice the shape. The shape is really specific. If you get close in here, you'll see that there are lots of opportunities for this antibody to make very loose chemical bonds with the S protein. And in this case, we call the S protein an antigen, or AG, and the antibody is abbreviated AB. Okay? So that's an important interaction. And the reason it's important is because what we're starting to see in the world is some brand new strains of coronavirus where the S protein, this is the S protein still, looks like this. Notice the difference. This shape will no longer fit into this in a perfect way. So when we have this, all of a sudden, there's no binding that takes place here, and this antibody does not recognize this protein. All right, so that can create the potential for problems down the road, which is problematic, and we'll talk about that in just a second. Okay, so let's back up away from this then and try and figure out why antibody to S protein is so important, and why is it that scientists chose this protein to be in the vaccine. Okay, so the reason they chose the S protein is this. This virus floats around in the air, it lays around on surfaces, and it comes into contact with our bodies either through our mouth, our nose, or our eyes. Those are the three primary places of transmission. Inside your mouth, your nose, and your eyes, there's a layer of cells. And these cells are squamous epithelia, and yes, I know the scientists in the room will say those don't look like squamous epithelia, but just trust me, for these purposes, they are. And squamous epithelia have receptors on them that this virus recognizes. So interestingly enough, the receptor looks almost exactly like this arm over here with the antibody. And so the receptors are recognized by the virus, and the virus can gain access to the cell, and it actually penetrates the cell. So what happens with this virus is once the cell membrane merges, it releases its little pieces of RNA into the cell, and then this RNA takes over all the machinery inside the cell and starts making new viruses. So it cranks out lots of new virus particles, which are then secreted into the bloodstream, and they go and infect more cells. All right, so the antibody, or this protein was chosen, the S protein was chosen because it's the most important protein on the virus that binds to the cell. So if you can physically inhibit this, if you can cover these guys up with antibody molecules, then you can block this interaction. So I'm going to try really carefully just to erase this to make my point here. So if we can cover this up with an antibody, then you can see there's nothing to interact with, and that fusion becomes blocked. The virus can't gain access to the cell. All of this becomes blocked, and none of that happens. So that's why this S protein was chosen, because it's so critical in the life cycle of the virus. All right, so as important as that is, and it really is crucially important, it's also the weakness of this vaccine, because what we're starting to see is strains of virus emerging that have a different shape on their S protein. The one that I had up before and erased, we're starting to see things like the Lambda strain that have different spike proteins, and the antibodies are having a hard time recognizing them, which means that at some point, the Pfizer and the Moderna vaccines are going to have a difficult time dealing with certain strains of this virus. Okay, I'm going to pause there and erase this and come back. We're going back just a little bit here, but I want to make sure we understand something. So you'll remember that when we talked about the primary immune response that looks like this, that there's this lag period. And the lag period can be anywhere really from 3 to 10 days or so between infection, which is here, and the onset of symptoms, which is here. And it's during this time, sometimes there's virus shed and people are really contagious, and the whole point of having a vaccine is so that you can mount this immune response before you actually get disease. So you remember that we wanted to have the vaccine so that, in the second or third time you saw something, instead of this little peak, if you're at day 0 here, you're making this really large peak that comes out like this. So that's the whole point of getting the vaccine, so that if the real virus comes along, you've got a pre-existing immune response, and that pre-existing immune response is protective. That's what you're trying to get. All right, so now we've got a couple of questions that came in after the first video that I want to try to address. And one of the questions was this: if I've had COVID, why do I need to get the vaccine? And the answer is pretty simple, and the answer is this right here. Remember this is our spike protein, but then we also had a protein that looks like this, and I'm not naming these, though they do have names. We have other proteins that are present on the surface of this cell, or on the surface of the virus, that the immune system can recognize. So when this is a natural immune response and not a vaccine, we don't know in this first peak of IgM whether that IgM is directed against protein 1, protein 2, the S protein, or protein 3. We don't know. All right, but what we do know is that to prevent infection, this is the one that you need to block. So if you've had a natural infection with COVID and you mounted an immune response to protein 1 or 2 or 3, but not S, then you're still going to be able to be infected pretty easily by this virus. Okay, so the whole point of using the vaccine against S is to block that interaction. And I won't draw it again, but you remember the virus and the epithelial cell. We want to try to block that interaction. If you don't mount an antibody response to S, you can't block that interaction. So you might have mounted an immune response, but it's not protective. So the reason that we use this in the vaccine is that we know that this is a protective immune response. Well, then the question makes sense. If you've had the virus, if you've been infected and you've survived COVID, why do you get the vaccine? And the answer is really simple. It's this. The vaccine is an insurance policy. All right? You don't know if you had the natural infection, if your body made antibodies against this protein. But if you take the vaccine, the only thing in the vaccine is that. And so you'll know from the vaccine that you'll mount an immune response to that. So if you've already had COVID, getting the vaccine is not much more than an insurance policy, but it's a pretty critical insurance policy because it will allow you a variety of things that just give you that much more protection, including a pre-primed immune response, a whole set of T cells that are going to be able to recognize S protein, and all of the cytokines that are involved in driving this really large response should you actually ever see the virus again. Now, I want to take it the other way too. So what if you've already had the vaccine and you get one of those breakthrough infections? Because they do occur. Well, remember, I said breakthrough infections do occur and they will occur because this is IgM and this is mostly IgG. And what's protective on the mucosal surface? Well, it's this one, IgA, which there might be a little peak of IgA in here, but it's not very big. You don't see IgA usually until later on. So really the best way to approach COVID from an immune system point of view is this. If you get vaccinated, let's do this. Let's say you get vaccinated here, your first shot, you mount a nice little IgM response. You get your second shot over here. That's the second Pfizer or Moderna shot. You get a nice peak like this that's IgG. And then sometime out here, you're over visiting your friends at a party or whatever, and you actually contract COVID. Well, what's going to happen here? Well, firstly, you've already made an IgG and an IgM response, so your immune response is going to shoot up like this. I mean, it's practically vertical in terms of the amount of antibody that you make. All right, but what's going to happen in this response is the tail goes much further, and what we call differentiation to IgA goes up dramatically when you actually see COVID for the first time. So this is going to be much more protective over a longer period of time if you get the actual infection here. And there's a second benefit. All of this immune response that you've made heretofore is all directed against that spike protein. But if you actually get COVID now, you've got a dominant spike protein, but you'll start to make antibodies to these too. And you'll start to make antibodies to the round one, like this. And you'll start even to make antibodies to this little squiggly one down here. So very quickly, you've got these nice antibody responses to a wider variety of surface proteins, and that gives you even better protection out here. All right, so the folks that have been vaccinated twice and then get COVID are probably among the most protected, immunologically speaking, from serious repercussions from the virus. Now, as I said in the very beginning, you remember that we all respond to these things on a bell curve. And there are a group of people down here who will not make strong immune responses no matter what happens here. Generally, these people are over the age of 75, or they have some serious underlying health conditions that prevent their immune responses from being mounted in the first place. The folks out here, they're just making crazy wild immune responses, and they're probably not even going to have apparent COVID infections over here. The rest of us all operate in this curve, and we would do extraordinarily well to get the vaccine first, and then if we have a breakthrough infection with actual COVID, we'll mount really strong protective responses. Now, there's one more thing that I want to mention, and it's based on some really early research that's starting to come out now, and that's this. We have identified a new protein here on the virus, and the name has just escaped me, but this appears to be the protein that mediates fusion with the host cells, with our epithelial cells. And it seems to be buried a little bit in the membrane. It's an insertion protein. I knew it would come to me in the end. So this insertion protein is interesting. We don't seem to mount very good immune responses to it, certainly not with the vaccine, with the S protein, and we're not sure about how the body reacts to it in its native form like this. But there's at least one paper that's been published now that if we can isolate this insertion protein and put it in a vaccine, there is a good chance that it'll be protective against all strains of the COVID virus. Now, I say that with some hesitation because there'll be some people who are listening to this who say, whoa, I'll wait for that one. When that one comes out, then I'll get vaccinated. I wouldn't recommend that because you may get COVID before that one comes out and it can kill you. So what I would recommend is get your first shot, get your second shot. If you get COVID, most of the time this is a really mild form of disease. And in this response, you may be making your own antibodies against this insertion protein. But this holds out some hope for the future because what we're seeing right now in terms of variants of the virus is that we may not have enough letters in the Greek alphabet. There are a large number of variants of the virus that are coming out, and many of them have S proteins that are different. So we don't want to be caught in that cycle forever where we need to constantly vaccinate against the S protein of SARS-CoV-2. What we'd like to do is figure out a vaccine that protects against all strains of COVID. And at least now, and again, only one paper, that's all I've seen, one paper points to this insertion protein as a potential marker for something that might be a universal COVID vaccine. So a little hope there, but for now, as I said last time at the end of the video, get vaccinated. Thanks for watching.